مساء الخير. احنا النهاردة هنتكلم عن chapter 6 اللي هو chapter ال RNA Purification. في الشابتر ده احنا هنتكلم عن confirmation of RNA integrity, total RNA isolation techniques, preparation of DEPC treated solutions, RNA extraction from blood, RNA extraction from frozen tissue, RNA extraction from tissue sections. طيب احنا هنبدأ بأول حاجة الانترودكشن. RNA like DNA contains a sugar phosphate backbone and nitrogenous bases. The major difference are that the sugar is ribose, the base is uracil incorporated in place of thymine, and the molecule is single-stranded. Total cellular RNA is a mixture of three types: transfer tRNA, ribosomal rRNA, and messenger mRNA, which is a template for protein production. Initial transcription results in the formation of a primary transcript that includes introns, exons, and the untranslated region. Exons are the segments that encode the amino acid sequence of protein. Introns are intervening sequences that don't encode a protein, but guide splicing of the mature mRNA and allow alternative combinations of exons to produce variation of similar proteins. Excision of introns and splicing of exons must occur before mRNA protein translation can occur. One of the first decisions that the researcher has to make when detecting or quantitating RNA is whether to isolate total RNA or poly-A selected RNA, also commonly referred to as mRNA. This choice is further complicated by the confusing array of RNA isolation kits available in the marketplace. In addition, the downstream application influences this choice. From a purely application point of view, total RNA might suffice for most applications, and it's frequently the starting material for applications ranging from the detection of mRNA species by nursered hybridization to quantitation of a message by real-time PCR. The preference for total RNA reflects the challenge of purifying enough poly-A RNA for the application. mRNA comprises larger than 5% of cellular RNA. The potential loss of a particular message species during poly-A purification and the difficulty enough um, and the difficulty in quantitating small amounts of purified poly-A RNA if the data generated with total RNA don't meet your expectations, using poly-A RNA instead might provide the sensitivity and the specificity that your application requires. The pros and cons with either choice are discussed below. Your experimental data will provide the best guidance in deciding whether to use total or poly-A RNA. Two situations where using poly-A RNA is essential are cDNA library construction and preparation of labeled cDNA for hybridization to gene arrays. To avoid generating cDNA libraries with large numbers of ribosomal clones and non-specific labeled cDNA, it's crucial to start with poly-A RNA for these procedures. دلوقتي هنتكلم عن ال confirmation of RNA integrity. The integrity of your RNA is best determined by electrophoresis on a formaldehyde agarose gel under denaturing conditions. The samples can be visualized by adding 10 mg per milliliter of ethedium bromide to sample before loading on the gel. Compare your PREP's 28 subunit rRNA band located at approximately 5 kilobase pair in most mammalian cells to 18 subunit rRNA band located at approximately 2 kilobase pair in most mammalian cells. In high-quality RNA, the 28 subunit band should be approximately twice the intensity of the 18 subunit band. The most sensitive test of RNA integrity is Northern analysis using a high molecular weight probe expressed at low level in tissues being analyzed. However, this method of quality control is very time consuming and not necessary in most cases. Northern analysis is not tolerant of partially degraded RNA. If samples are even slightly degraded, the quality of data is severely compressed. For example, even a single cleavage in 20% of target molecules will decrease the signal on a Northern plot by 20%. Nuclease protection assays and real-time PCR analysis will tolerate partially degraded RNA without compromising the quantitative nature of results. دلوقتي هنتكلم عن total RNA isolation techniques. عندي 3 techniques. أول واحدة guanidinium cesium chloride method. الميثود دي slow laborious procedure but RNA is squeaky clean, unsuitable for large sample numbers, little if any genomic DNA remains. This method employs guanidinium isocyanate to lyse cell and simultaneously inactive ribonucleases rapidly. The cellular RNA is purified from the lysate via inactivate ribonucleases rapidly. The cellular RNA is purified from the lysate via ultracentrifugation through cesium chloride or cesium trifluoroacetate cushion. Since RNA is more dense than DNA and most proteins, it pellets at the bottom of the tube after 12 to 24 hours of centrifugation at more than 32,000 RPM. This classic method yields the highest quality RNA of any available techniques. Small RNAs, example 5 subunit RNA and tRNAs, can't be prepared by this method, as they will not be recovered. The original procedure were time-consuming, laborious, and required overnight centrifugation. The number and the size of samples that could be processed simultaneously were limited by the number of samples of spaces in the router. Commercial products have been developed to replace this lengthy centrifugation with easier, less time-consuming methods. However, if the goal were to isolate very high-quality RNA from a limited number of samples, this would be the method of choice. دلوقتي هنتكلم عن الميثود التانية اللي هي single and multiple step guanidinium acid phenol method. الميثود دي faster, fewer steps, prone to genomic DNA contamination, somewhat um, cumbersome if large sample numbers are be processed. The guanidinium acid phenol procedure has largely replaced the sodium cushion. The guanidinium acid phenol procedure has largely replaced the cesium cushion method because RNA can be isolated from a large number of samples in 2 to 4 hours without resorting to ultracentrifugation. RNA molecules of all sizes are purified, and the technique can be easily scaled up or down to process different sample sizes. The single-step method is based on the propensity of RNA molecules to remain dissolved in the aqueous phase in a solution containing 4M guanidinium thiocyanate pH 4 in the presence of a phenol chloroform organic phase. At this low pH, DNA molecules remain in the organic phase, whereas proteins and other cellular macromolecules are retained at the interphase. دلوقتي هنتكلم عن الميثود رقم 3. الميثود دي اسمها non-phenol based methods. الميثود دي very fast, very clean. Clean RNA can process large sample numbers, possible genomic uh, possible genomic contamination. One major drawback to using the guanidinium acid phenol method is the handling and disposal of phenol, a very hazardous chemical. As a result, phenol-free methods based on ability of glass fiber filter to bind nucleic acid in the presence of chaotropic salts like guanidinium have gained favor. As with the other methods, the cells are first lysed in guanidinium-based buffer. The lysate is then diluted with organic solvent such as ethanol or isopropanol and applied to a glass fiber filter or resin. DNA and proteins are washed off and the RNA isolated at the end of aqueous filter. At the end in an aqueous buffer. This technique yields total RNA of the same quality as phenol-based methods. DNA contamination can be higher with this method than phenol-based methods. Since DNA are column-based protocols requiring no organic extraction, processing large sample numbers is fast and easy. It is also among the quickest methods of RNA isolation, usually completed in less than 1 hour. التوبيك اللي بعد كده inhibition of RNAs. The most common method of preparing RNase-free solutions is diethyl carbonate (DEPC) treatment. DEPC inactivate RNAs by carboxy acylation of a specific amino acid side chain. Specific amino acid chain in protein. DEPC is a suspected carcinogen and it should always be used with the proper precautions. دلوقتي ال preparation of DEPC treated solutions. Most protocols specify adding DEPC to solutions at the concentration 0.1%, followed by mixing and room temperature incubation for several hours to overnight. The container lid should be loosened for the extended incubation because a considerable amount of pressure can form during the reaction. Finally, the solution is autoclaved. This inactivates residual DEPC by hydrolysis and release CO2 and ETOH as byproducts. The ATOH byproduct can combine with trace carboxylic acid contaminants in the vessel to form volatile esters, which impart a slightly fruity smell to the solution. This doesn't mean that trace DEPC remain in solution. DEPC has half-life of 30 minutes in water, and at DEPC concentration of 0.1%. Solutions autoclaved for 15 minutes per liter can be assumed to be DEPC free. Be aware that increasing the concentration of DEPC to 1% can inhibit more RNAs, but can also inhibit certain enzymatic reactions. So more is usually not better. DEBC treatment of glassware distilled water. Add 0.1% DEBC to distilled deionized water and fill glassware and leave to stand overnight. Decan the water and autoclave DEBC treated water and sterilize glassware at 220 Celsius for 2 hours. Drive DEBC off by both procedures. RNA extraction from blood. As with any RNA protocol, extreme care should be taken to exclude RNA's contamination, the greatest source of which will be the sample itself. All disposables and reagents should be RNAs free. Materials: microfuge tubes 1.5 milli, ice bucket, microfuge, red cell lysis buffer 1.6 sucrose, 5% triton, x minus 100, 25 m MgCl2, 60 m tris HCl, pH 7.5, stored at 2 to 8 Celsius and used cold. Extraction buffer 5.25 m guanidinium thiocyanate, 50 m tris Cl, pH 6.4, 20 m edita, 1% triton x minus 100, 0.1 m beta mercaptol ethanol. Add immediately. Prepare to use. 2 m sodium acetate, pH 4, phenol saturated with 1 m tris HCl, 0.1 m edita, pH 8. Chloroform isoamyl alcohol 24:1. Isopropyl alcohol 70% ethanol, RNase-free distilled water. Method: in a microfuge tube, mix 100 microliter anticoagulated blood with 1 milli of red cell lysis buffer. Leave at room temperature with occasional shaking until the red cells have lysed the solution translucent, usually within 5 minutes. Microfuge for 3S at 13,000 gram to pellet the white blood cells. Remove and discard supernatant. Add 200 microliter of extraction buffer and resuspended cell pellet by drawing through narrow needle several times. Add 20 microliter of 2M sodium acetate and mix gently by inversion. Add 220 microliter of phenol and mix gently by inversion. Add 60 microliter of chloroform isoamyl alcohol 24:1 and vortex. Place on ice for 15 minutes. Microfuge at 12,000 gram for 5 minutes and transfer the upper phase to new microfuge tube. Add 200 microliter of ice cold isopropanol mix and store at minus 20 Celsius for 30 minutes. Microfuge at 12,000 gram for 15 minutes and discard supernatant. Resuspended pellet in 200 microliter of extraction buffer. Repeat 3 steps through 9. Wash pellet with 400 microliter of cold 70 ethanol. Microfuge at 12,000 gram for 5 minutes and discard supernatant. Carefully remove last traces of ethanol from tube. Resuspended in 100 microliter of distilled water and incubate at 50 Celsius for 15 minutes to dissolve RNA. RNA extraction from frozen tissue. RNA extraction is fundamental to all aspects of mRNA analysis. We include here a simple method that avoids use of mortar and pestle materials. All chemicals, unless otherwise noted, were molecular biology grade. All glassware was pretreated with diethyl carbonate (DEBC). All deionized distilled water was pretreated with DEBC and autoclaved DEBC water. DEBC is a potent anti-RNase agent. Prone microdismembranator and Teflon vessels. 3M lithium chloride, 6M urea, dissolve in 800 milliliter of DEBC water and make up to 1 liter. The solution can be stored at 4 Celsius for 3 to 6 months. 10 millim trace HCl, 0.5% sodium dodecyl sulfate, SDS, pH 7.5. Prepare stock solution of 10% SDS and 0.5 m trace HCl, pH 7.5 in DEPC water. Stocks are stable at room temperature for up to 12 mO. Proteinase K, prepare 1 gram per milliliter W per V DEBC water stock and store at minus 20 Celsius for up to 12 months. Dilute in 10 m trace HCl, 0.5% SDS as required. Discard unused diluted enzyme. Phenol chloroform isoamyl alcohol. Phenol is pre-saturated with 10 m trace HCl, pH 7.5. Prepare a mixture of 25 to 24 to 1 phenol chloroform isoamyl alcohol. Store at room temperature for up to 6 months. Shielding from light. Method: tissues should ideally collected fresh and stored in liquid nitrogen. Routinely, samples are collected on ice and transported for freezing within 30 to 60 minutes. Tissues are disaggregated using prone microdismembranator. Teflon vessels and steel ball bearings are cooled in liquid nitrogen before use. Frozen tissue 500 mg is placed in the vessel with a single ball bearing and agitated at 1000 cycles per second for 60 seconds. The vessel is then recooled in liquid nitrogen. This process is repeated until tissue is powdered. Immediately after disintegration, of tissue material is resuspended while frozen by adding 1.5 milli of lithium chloride per urea and transfer to separate tubes. The vessel is washed further to X with 1.5 milli of lithium chloride per urea and washing combined with original sample. The resuspended medium is made up to 6 milli in lithium chloride urea and sonicated 4 to X30S at maximum power using probe sonicator. The sonicated samples are stored overnight at 4 Celsius. Centrifuge at 15,000 gram 4 Celsius for 30 minutes. The supernatant is discarded and the pellet washed with further 6 milli of lithium chloride per urea. Recentrifuged 15,000 gram for Celsius for 30 minutes and the supernatant again discarded. The pellet is resuspended in 6 milli of trace HCl per SDS with 50 milli proteinase K and incubated at 37 Celsius for 20 minutes. Samples are extracted with 100% phenol, followed by phenyl chloroform isoamyl alcohol. After each extraction, the sample is centrifuged at 2000 gram at room temperature for 10 minutes, and the aqueous phase is recovered. After the final extraction, 300 microliter of 8 lithium chloride and 2.5 volumes absolute alcohol are added and the sample restored at -20 Celsius for 30 minutes overnight. RNA is pelleted by centrifugation at 4000 gram for Celsius 45 minutes. The supernatant is discarded and the RNA pelleted dried and resuspended in DEBC-treated distilled water. Concentrations are estimated by optical density at 260 per 280 nanometer. RNA extraction from tissue section. There are two different methods of preparing tissue for histology. Paraffin embedding and freeze embedding. Each has their advantages and drawbacks. Paraffin embedded tissues produce optimum morphology but have comparatively poor molecular preservation and recovery. Although frozen sections have poorer histology, they allow excellent recovery of DNA and RNA for analysis. Although fixation is performed to preserve the morphology of the living tissue, it doesn't necessarily have a beneficial effect to RNA and DNA. Formalin, one of the most popular fixatives, cross-links nucleic acids to protein, thus making the molecules rigid and susceptible to mechanical shearing. The duration of formalin fixation also seems to be important. Studies that have demonstrated DNA recovery around 200 base pair recommended a period of fixation from 16 to 24 hours, but not any longer. RNA is more liable species, and the paraffin embedding process has been shown to greatly harm it. Many studies have shown that formalin fixation has the worst effects among commonly used fixatives, and the ethanol-based fixative as having the best RNA preservation. Materials: microfuge tube 1.5 milli, ice bucket, microfuge, extraction buffer 5.25 m, guanidium thiocyanate, 50 m trace HCl, pH 6.4, 20 mm, EDTA 1%, Triton X minus 100, 0.1 m beta mercaptol ethanol. Add immediately before use. Glycogen 8 mg per milliliter in distilled water, 2 m sodium acetate, pH 4, phenol saturated with 1 m trace HCl, 0.1 m EDTA, pH 8, chloroform isoamyl alcohol 24 to 1, isopropyl alcohol 70% ethanol, RNase-free distilled water. Method: the extraction is initiated by incubating tissue sections or micro-dissected cells in 500 microliter extraction buffer for 5 minutes at room temperature with gentle agitation, inverting several times. Add 20 microliter of 2 m sodium acetate and mix gently by inversion. Add 220 microliter of phenol and mix gently by inversion. Add 220 microliter of phenol and mix gently by inversion. Add 60 microliter chloroform isoamyl alcohol 24 to 1 and vortex. Place on ice for 15 minutes. Microfuge add to 12,000 gram for 5 minutes and transfer the upper phase to new microfuge tube. Add 1 to 2 microliter of glycogen 10 milligram per milliliter. Glycogen is a carrier that is used if RNA quantities are less than 1 microgram. It also facilitates visualization of the pellet. Add 200 microliter of ice-cold isopropanol mix and store at -20 Celsius for 30 minutes. Microfuge at 12,000 gram for 15 minutes and discard supernatant. Resuspended billet in 200 microliter extraction buffer. Repeat step 3 through 9. Wash pellet with 400 microliter of cold 70% ethanol. Microfuge at 12,000 gram for 5 minutes and discard supernatant. Carefully remove last traces of ethanol from tube. Folded sterile. Swab. Resuspend in 100 microliter of RNase-free distilled water and incubate 50 Celsius for 15 minutes to dissolve RNA. ودي الريفرنسز اللي احنا أخدنا منها المعلومات عن ال RNA Purification.